out. Okay, so we're starting on that one. Are you ready, Dakota? Okay, so you should know proteins number 61 are made of amino acids. The building blocks of proteins are amino acids. You should be able to give me the jobs of amino acids or proteins. Okay. The sketch number 65, you also will have that in your labeling. We'll go over that. Okay. Amino acids are linked together with dehydration synthesis number 66. Number 67, many proteins can be enzymes or biological catalysts. Jump down to number 73, protein chains are called polypeptides. Okay. Denaturing a protein can change its pH. Two things that can denature the protein, like cooking, so heat. So temperature, pH, and go ahead and add concentration to that. The concentration of things can also, when we talk about enzymes, we're doing this on Friday, by the way. You'll get to do this last, okay? Go ahead and highlight about the hemoglobin in your red blood cells. That's an example of a protein in your body. You also have insulin in your body. You also have proteins in your cell membrane that are receptors. Those are good examples to use.
So do you just want like all of 79 is what you're saying?
Yeah, is that all 79? I said B, C, D, and E. Okay. Those are good examples of proteins found in your body that are useful. Let's look at your lab. Identifying fats and proteins, Dakota, and let me give you back yours, and then I'll take it back. On your lab, let's focus on the back page. Number 7. Number 7 is a good question. Tell me the test that you used to test for proteins. What was that stuff called? Burette reagent. Do you need to write that down? Go ahead and write that down. Burette, B-U-I-R-E-T, reagent. That was your what? What is that called when it changes colors? Indicator. So that's your indicator. You need to put that in parentheses, indicator. Go ahead and write that. So it was your indicator. And what color was burette reagent?
Blue.
It was a blue color. And what did you do with it? Tell me what you did.
Well, we added to the things that we thought had proteins, and if it turned purple, it's a protein.
That's right. So when you add the burette reagent to your test tube, if it had protein in it, it turned purple. Okay? You should be able to describe that in a lab or on a test, what you did in the lab. Okay? In terms of distribution, you should know, things like what had protein in it, things like your fingernail had protein in it, your hair had protein in it, but then things like the bean seed, it had protein, but it also had starch. So those are good examples to say most most of the things that we looked at though came from what? Consumers or producers? Consumers, like the egg white, right? The milk. Alright. Is there any questions that you have about the lab? Can you explain what to do in the lab? Yes. Okay. So let's put the lab aside and let's go to the squirrel page. Yeah, get that one out and turn to page 5. You need to know your amino and carboxyl group. Be able to identify those, so circle your pictures. The basic structure of an amino acid on page 6. Know that. Be able to tell me the parts. Be able to tell me that that whole thing is a basic amino acid. Now your side chain, your R groups, they can be different ones. But the functional groups that are always the same in these are going to be the amino group, the NH2, and your carboxyl group, the COOH. Okay? I think we're okay on this one. We've covered the pictures are going to be the big thing on that one. So that's a good one to look at the pictures for. Okay? Not that you can go back over your questions on your own. The other one, chemistry of fats and proteins. That's it. Okay? We highlighted a lot on this one already. Number 22. Do you remember the rule? What did I tell you yesterday? If you link 6 amino acids together, how many water molecules do you make? 5. So it's always one less. So there'll be probably a question about that on the test. So if you're not sure, make sure that you look back at number 22. Number 8. No, number 9. We use the same thing. You use dehydration synthesis to make a protein, right? To link the amino acids together. So you're taking away the water molecule. But to break the molecule, the protein molecule apart, what do you need to do?
You have to, you have to do hydrolysis.
Hydrolysis. So highlight number 9. The key word there is hydrolysis. It should be in italicized. That hydrolysis means you are adding the water back in to break the protein molecule apart. Okay? Are there any questions about that? The last column on the chart on amino acids, those are good general true false type questions I may ask, or yes no type questions. Okay? And that should be it. Okay?
We'll do better on
There are 16 questions. There's matching, true, false, yes, no type questions. A couple multiple choice, and then typing out. Where you guys do the best seems to be the typing out. You got, yeah, you do. You're able to.
Not for me.
Many of you do really well on explaining.